Okay.
Hello, everybody. Welcome to another episode of our Western Biology Evolution Seminar Series. I'm your host, Matheus Sanita, a PhD student here at the Department of Biology at Western University. And today I have here with me Dr. Andrew Doxey, and I have a short bio for you guys. So Dr. Andrew is a bioinformatician and associate professor in the Department of Biology at the University of Waterloo, where he holds a university research chair position. He's cross-appointed to the Cheriton School of Computer Science, and he's also an adjunct professor in the Department of Medicine at McMaster University. The Doxey Lab focuses on bioinformatics, microbial genomics, and molecular evolution, and they apply computational methods to discover new protein families and functions. Recent work includes the development of the NO3 phylogenomics database and studies on the evolution and diversity of bacterial toxins, such as botulinum, diphtheria, and C. difficile toxins. Dr. Doxey is supported by grants from NSERC and Mitex, and is the recipient of the 2018 Thermo Fisher Award for contributions to microbiology. Andrew, thank you very much for being here with us, and I would say the floor is all yours.
Okay, well, thanks so much for having me. I'm happy to present this work. This just came out a couple of weeks ago, so I'm excited. Today, I'm going to talk to you about a recent project from our lab looking at ancient DNA, and particularly looking at a pathogen known as Clostridium tetani, the tetanus microbe in these ancient samples, and how we used genomics to investigate it and identify novel toxins. Just a bit of background into my lab. So I run a bioinformatics lab at the University of Waterloo, and we're quite generally interested in a number of different areas in bioinformatics, everything from the development of novel computational tools to doing genomic data mining to discover new protein families and functions. We're also really interested in computational metagenomics and also evolutionary genomics and pathogen genomics. We've, over the last 5 years or so, we've really been focusing heavily on identifying pathogens within complex sequencing data sets, looking at variants of pathogens and their virulence factors, and trying to use computational methods to detect important differences and adaptations within pathogen genomes. And so today I'm going to talk about a recent project that really combines a number of these different areas together, and that is this work I told you about already, looking at ancient Clostridium DNA associated with human archaeological samples, and this was just published. So check this out if you're interested. It'll have a lot of details that I can't get into today. So I'm just going to give a really quick background on this pathogen. A very familiar pathogen, Clostridium tetani. It's familiar because this is an organism that has plagued humanity for thousands of years, but one that it is also that is also a target of common vaccines. Okay, so we're all vaccinated against tetanus toxin. We're all familiar with the scenario shown here on the left, where if you are not vaccinated and develop a wound, then spores from C. tetani can infiltrate your body, and it's really the toxin itself, which is called the tetanus neurotoxin, that alone is responsible for causing the disease phenotype. Okay, so very briefly, what this neurotoxin does, it is similar to botulinum neurotoxin. Actually, it's the second most potent toxin that has ever been characterized. And what it does is it is capable of entering host inhibitory motor neurons. And once it enters those neurons, it's able to proteolytically cleave specific proteins called snares. And in doing this, this protease-mediated cleavage of snare proteins, what that does is it ends up inhibiting the neurotransmitter release from these neurons, which ends up resulting in spastic paralysis. Okay, so it's part of a general family of toxins called clostridial neurotoxins. Again, botulinum neurotoxin is another form of it, which causes botulism and acts in a bit of a different way and targets different neurons. Now, the biology of C. tetani is very well understood. And as I mentioned, it's a disease that's been preventable by vaccination. However, the evolution, the origin, and also the diversity of this organism is not well understood. And this has actually been an interest of our lab, not just looking at Clostridium tetani specifically, but looking at neurotoxigenic Clostridia. Okay, Clostridium species that produce these highly potent neurotoxins. It's a really interesting question to try to understand where these species came from, where the toxins came from, how they evolved, perhaps what other hosts or other ecological origins these species might have. So just a little bit on what's known about the genomics of C. tetani. So C. tetani possesses a chromosome and also a plasmid. And it's the plasmid that encodes the virulence factors, particularly the tetanus neurotoxin is encoded on that plasmid. And genomes are available for 38 modern strains. And I'm showing you a tree, phylogenetic tree of those genomes on the left. You can see that they generally split into two major clades, clade 1 and clade 2. And there's actually quite limited genomic variation between these different strains. In fact, if you look at the tetanus neurotoxin itself, it's extremely conserved. There's very little divergence or variation that's seen at the sequence level between different neurotoxins from 90, they're 99 to 100% identical. The earliest strain actually dates back to to 1920 or so. This is known as the Harvard E88 strain, and this is actually still used as the reference genome or the reference strain for C. tetani today. Now, despite that, though, there are written accounts of tetanus disease, or at least tetanus-like phenotypes, symptoms that date back to ancient times. They go back as far as the ancient Egyptians through written descriptions, as shown here. I'm not going to read this in full, but this is coming from 1650 to 1550 BCE, and the written description matches that of what's known as lockjaw or tetanus. There are also written descriptions in the ancient Greek literature through Hippocrates and also ancient Roman literature as well. So it's understood that tetanus as a disease has been around, has affecting humanity for quite some time, and so very likely it's an ancient disease, and it probably has its origins in as an animal disease that co-evolved, right, with different species. I should mention that tetanus is also observed in animals, but again, this jump and specificity for different hosts has not really been explored in great detail at a genomic level, or an evolutionary level as well, I should say. Now, one of the really exciting things that has really emerged in studies of ancient pathogens and ancient microbes is the use of ancient DNA. And so ancient DNA can be collected from archaeological specimens. This is just a really general overview. You can start with a bone or a tooth sample, for example, and extract DNA within these samples. And depending on where the extraction takes place, you will get differing amounts of endogenous DNA that really dates back to the ancient time period of that sample versus more modern contaminant DNA. I'm not going to get into that, but what's done generally is DNA is extracted and it is sequenced using modern approaches. Okay, so high throughput sequencing, just as you would modern DNA. There are some differences as well. So there's a chemical procedure that you can apply to try to remove some of the ancient damage that happens with highly fragmented and mutated ancient DNA molecules, but I won't get into that until a little bit later. But one of the exciting things is that this ancient DNA contains not only human-derived ancient DNA, from which you can do a lot of different analyses of these ancient specimens, but also it contains a mixture of ancient microbial DNA that may have colonized that sample, or maybe have been present in the ancient specimen. And this can be analyzed through metagenomic type methods. So you can take standard methods that we apply to modern metagenomic samples, and we can use those to analyze these ancient DNA data sets as well. And for example, one can take the individual reads and assign putative taxonomy to each of those reads. We can compare them to the database of microbial genomes that we have available now, and we can try to assign them their taxonomic origin. Okay, and so we can look at different samples, like here on the right, sample A, B, and C, and we can get an idea of the taxonomic composition of these samples. And we find one of the really interesting things about ancient DNA is that the majority of the DNA that's found in a lot of these samples, for example, a lot of tooth samples, is of a microbial origin, not a human origin. And for a long time, it was suspected that this DNA fraction that was microbial was contamination. A lot of studies, however, have shown that that's certainly not always the case. Some groundbreaking studies of ancient plague, Yersinia pestis, Mycobacterium tuberculosis, have shown that no, you can identify genuine ancient pathogens within these samples as well. And a major goal of bioinformatics is to try to distinguish these sort of genuine ancient DNA sequences from ancient pathogens, from contaminating organisms. Now I want to talk a little bit about the source of the data set that we've really looked at in this study, and that is the NCBI Sequence Read Archive. So the Sequence Read Archive contains really the raw sequencing data that for millions of different samples. This is not GenBank, this is not assembled genomes, but this is the raw DNA fragments that are in for example, FASTQ files that accompany genome projects and metagenomic sequencing projects, RNA-seq projects, many different things. And the NCBI SRA contains an enormous amount of data. This is probably a little bit out of date now, but at the time of our analysis, it was 26 petabytes of sequencing data. And like I said, from any type of sequencing project, and one of the exciting things is that there's a lot of data in the SRA from these ancient DNA or archaeological sequencing projects. And so that allows us to actually do broad bioinformatic surveys of ancient DNA. We can look in ancient DNA, not necessarily only in a targeted way, looking at specific samples, but we can do bioinformatic screens to see if we can find DNA that is related to organisms of interest as a survey across many different ancient DNA data sets and also modern data sets as well. And that's really what started this project, and I'm going to talk to you about it. We wanted to use publicly available data in the SRA to do a very broad, large-scale search for this organism that we were interested in, Clostridium tetani. Now, just to tell you a little bit about the bioinformatic methods here. So many of you may be familiar with tools like BLAST or other kind of related sequence alignment or database search tools like Diamond or MMSeqs. Okay, now these are incredibly powerful, but actually it's very challenging to apply tools like this on the scale of the SRA, Sequence Read Archive. There's an enormous amount of data there, and so these tools were not used for our study, and what we had to do, we actually had to look at these data sets in a slightly different way. And fortunately, there was another tool available. This is called STAT, and what STAT does is it actually pre-computes. It uses k-mers, okay, so short sequence fragments that map and that are predictive of taxonomy. Okay, and it uses those fragments to essentially estimate a rough taxonomic profile for any sample, and STAT was actually applied to all of the samples within the SRA database so that you could look at any sample you're interested in and get a rough estimate of the taxonomic composition of those samples. Okay, so for example, here's just a random SRA data set, and you know, okay, most of the reads in this particular case are not identified. They map to nothing in the database, but here we have almost 4% of the reads that can be matched through these short k-mers to reference organisms that are in our current database, and so that way you can get an idea of what taxa might be there. So we took advantage of that. We used that really as the starting point of our analysis, and we looked at over 10 million different data sets in the SRA from almost 300,000 studies. This is really an enormous amount of data, and we performed a screen. We ranked all of those data sets according to their predicted C. tetani abundance. We wanted to see which data sets out there have the highest levels of this organism that we're interested in, C. tetani. And this is just a visual depiction of our screen here. So what I'm showing you are each data point here is a sample within the SRA. And I'm plotting the data set size on the x-axis here, and on the y-axis I'm showing you essentially a count of the number of C. tetani affiliated reads that we're finding, or Kmers, I should say, that we're finding in these samples. And one of the interesting things is that so if we apply a very strict threshold, okay, we're only looking at the most extreme cases, then we find an interesting mixture of samples. We find that, okay, about 28 of these samples are indeed derived from or are associated with C. tatani genome project. So these are kind of controls in a way. This is what we expect, and that was good to see. But in addition to that, we also see many other samples that look very interesting. 29 of these are non-human samples. The rest are human samples. Only three of them were from, these are actually from human gut metagenome projects, but completely unexpectedly, 76 of these samples came from ancient DNA studies, and this was not something we expected to find at all. So these are archaeological samples, these that are associated with human human bone and tissue and tooth samples, and never had were these analyzed for C. tatani before. Okay, so that was not the original intent of these studies. Here I'm showing you just a geographical distribution of those samples. So they span quite a large time frame. The oldest archaeological samples date back to 4000 BCE. And we have also some more recent samples coming from sort of the Middle Ages, and lots in between as well. Geographically, they're also quite diverse. So quite a few of these samples around this time frame here are derived from, as I say, the Middle Ages. These include plague cemeteries from Europe. Okay, we have the Jomon shell mound sample here, dating about 1044 BCE in Japan. Interestingly, there are a number of ancient Egyptian mummy samples as well, where we're detecting high levels of C. tatani. And in this region of Chile, there are also these samples here that I'm going to highlight. These are going to come up a bit later. These are the Chinchorro mummies that date back to approximately 4000 BCE. These samples are of particular interest because these are actually the oldest known mummies. These predate Egyptian mummies by up to 2000 years. So these are really fascinating samples derived from ancient Chile. And again, what we're detecting is potentially high levels of this pathogen C. tatani in these samples. So the first thing that we wanted to do then bioinformatically is really verify that these Kmer-based taxonomic predictions were real or genuine, that we could really find additional evidence of C. tatani within these samples beyond just short fragments. And so what we did was we took the original data sets and we mapped the reads to the C. tatani chromosome that's on the left, okay, and also the plasmid that's shown on the right. So each ring here shows you the coverage. This is essentially the number of reads that are getting mapped to different parts of the chromosome, and the reference chromosome is shown on the outside versus the plasmid on the right. And so I'm showing you all 38 samples and sort of how much coverage we can detect for different parts of the chromosome. And you can see that it varies quite a bit, but even more so for the plasmid. That is, the plasmid, the coverage that we're detecting varies much more substantially. In some cases, we don't detect much of the plasmid at all. For these samples here, let me just get my laser pointer. Okay, so for these samples up near the top, we're detecting parts of the plasmid, some of the genes, but for example, the tetanus neurotoxin here is not being detected in these samples. And yet for all these ones below, you can see that we're detecting the tetanus neurotoxin here. This is another virulence factor called collagenase, and we're also detecting that gene quite broadly. But I do want to say that by detecting the tetanus neurotoxin gene, that in itself is indicative that we are in fact detecting C. tetani, because that gene is highly specific to C. tetani. There's no other organisms that have been found or identified that contain that gene. So it's very much a signature of C. tetani. I'm just showing you more here that of the taxonomic composition of these data sets. Okay, so these are the 38 ancient DNA samples, and up at the top, this shows you that a large proportion of the DNA does come from human, as you would expect. But there again, you could see that C. tetani is actually perhaps the most frequent organism that's consistently detected across these samples, as high as 75% in some. There are other microbes that are detected as well. There are a lot of other pathogens. For example, we've got Yersinia pestis in a couple samples, which isn't surprising for those, because they were actually these were plague samples, and there are associated studies that are done specifically on Yersinia pestis for those samples. But we also detect some unexpected things in these data sets that were not, again, the original intent of those studies. But what we wanted to then do is we wanted to use metagenome assembly methods to start piecing together the genomes of these ancient pathogens, and to see if we could build up larger contigs where we could do additional and more high resolution analysis. Okay, so here short reads are analyzed through metagenome assembly, okay, to get larger contigs. Those contigs are then analyzed using other methods to get more accurate identifications of taxonomic origin. And then what we can do is we can separate out, we can bin out those contigs that we are very certain are derived from C. tetani. So you can take these contigs, you can blast them against the reference database and ensure that their closest match is C. tetani, and even you can ensure that they are within a certain percentage identity range. Okay, so this almost guarantees that you end up with a highly accurate pool of contigs that are C. tetani. Okay, and so here I'm showing you now the same analysis as before. This is looking at all of the 38 samples, but in terms of the contigs themselves. We're also coloring each data set here based on the average percentage identity of the sequences to modern C. tetani. And what's interesting here is you can see quite a range where some of the ancient, or I should say the genomes associated with ancient samples, some of them are very closely related to C. tetani, high 90s in terms of percentage identity. But then again, we see a subset of genomes down at the bottom here whose average identity is more around the 90% range, which actually falls outside of the range of identity used to assign species. Okay, so generally we're talking sort of 95% plus for genomes within the same species. So this actually indicates that for some of these samples, we may not be detecting C. tetani, but actually potentially something related but distinct at the species level. This is an overview of, again, the 38 ancient draft genomes or metagenome assembled genomes that we've produced from these samples, and I'm showing you the predicted chromosome content, the C. tetani plasmid content, the fraction of the collagenase and tetanus neurotoxin gene that we're seeing in those samples, and also an estimate of the genome completion and contamination. And again, you can use metagenomic methods to estimate how complete is your assembled genome. And you can see that that some of the samples are very poor. Other samples are actually quite good in that regard. But there is some contamination, and we're dealing with mixed ancient DNA samples that might have complex microbial communities there, right? And it's also possible that there may be not just one strain of the organism you're interested in, but multiple strains, which can again complicate these bins or these metagenome assembled genomes that you're trying to build. The next question that we had, and this was a key one, is, well, how do we know that whether these genomes that we're identifying, this ancient C. tetani DNA, how do we know if it's ancient or if it's modern? And it's certainly conceivable that a lot of this DNA may be modern and may be derived from soil contamination or other types of contamination. Well, fortunately, there is an analysis that can be done to help distinguish modern and ancient DNA from these types of data sets. And that is by looking at the reads and looking for something called misincorporation rates or damage patterns within the reads themselves. Now, because of cytosine deamination, what happens is that ancient DNA tends to have more errors that accumulate near the edges of reads than modern DNA. So if you look at modern DNA and you observe the frequency of errors, okay, so for example, you expect an A in a position, but you observe a T, okay, then you kind of see a homogeneous or flat curve across for those rates across the molecule, okay, going from 5' to 3'. However, if you look at ancient DNA, one of the interesting things that you observe is that there's an accumulation of these misincorporations, these errors that occur at the edges of reads. And that has been shown to be really a hallmark, a distinguishing hallmark of ancient DNA. And in fact, the actual level of damage that's observed at the edges of reads can sometimes be used or a threshold can be used to distinguish ancient and modern DNA. And there's some debate over what that threshold should be, but one of the common thresholds is 10%. So if you observe 10% of errors at the 5 or 3' end, that or a damage pattern at that level, that can signify ancient DNA. Okay. The other thing that's worth mentioning is that it's also possible to inhibit this type of damage by treating your DNA sample with an enzyme, uracil DNA glycosylase, or UDG. And that enzyme, what it does is it cleaves deaminated cytosine residues. So it actually prevents the conversion, the incorrect conversion to misincorporation that happens during sequencing. And I should mention that is sometimes done in these ancient DNA studies in order to reduce the levels of damage in the molecules and obtain more accurate sequences. So we applied this kind of analysis to all 38 of our samples, and we saw some very interesting things. So first of all, on the left here, I'm showing you distributions of damage rates for different types of samples. As expected, if you look at modern C. tetani genomic samples, you do not detect damage, and this was done with a controlled data set of modern C. tetani genome projects. But if you look now at the ancient DNA samples that we have identified, you see quite a large range. Some of these samples actually have very high, or I should say, it's a relative thing, moderately high damage levels exceeding 10%, but there's quite a range. There are also some samples that are partially or fully UDG treated, and as expected, you see the damage levels for C. tetani in those samples, they decline. So this is the expected behavior. But it's really this group of samples where no UDG treatment was applied. This is where we see the greatest variation, and this is actually where we see some samples. There are 7 that exceed the 10% threshold. So this is looking like ancient C. tetani DNA in these samples. I should mention that it doesn't necessarily mean that the DNA in these samples dates back as far as the specimen itself, and it does not imply that this pathogen was responsible for the death of the individual in which associated with each sample. Okay, it could also have been post-mortem colonization by environmental microbes. Okay, and if that occurred even thousands of years ago, for example, for some of these samples, that might still leave evidence of DNA damage and that might still be ancient DNA. So we're not making any claims about that. We're just saying that the DNA is damaged and does appear to be ancient. These are the 5 top data sets in terms of their damage level. Okay, and we've named them based on the origin. And you can see there the damage pattern that is specifically occurring at the 5' and 3' edges of reads. Okay, so these are in red. These are C to T transitions or misincorporations, and at the 3', these are G to A misincorporations. So these are, again, errors that are accumulating because of cytosine deamination related damage at the edges of reads. I should also mention that on the left, I'm showing you the C. tetani damage. On the right, I'm showing you the damage patterns of human mitochondrial DNA that comes from the same samples. And one of the interesting things is that, so not only do we observe that there is human associated damage here as well, but we tend to see that the level of damage is higher for human mitochondrial DNA than for the C. tetani DNA, which may suggest that potentially the C. tetani DNA is less ancient, less damaged than the mitochondrial, or it might simply mean that the rates of damage differ depending on whether you're looking at a spore-forming microorganism versus a human mitochondrion. We looked at the damage patterns for all the samples, and one of the interesting things that we found is that actually there is a correlation between the C. tetani, so the ACBIN sense for ancient, Clostridium genomic or metagenome assembled bin. Okay, so if the damage for C. tetani correlates with the level of damage for human mitochondria. So this is encouraging, but again, as I mentioned, mitochondrial damage seems to be higher. There are some other factors that seem to be affecting the damage levels, whether capture methods are used. Okay, this is a type of sequencing where targeted sequencing where you use baits to pull out or enrich target microorganisms that you're interested in, or target genomes. We also see an influence of whether the samples were a mummy type or so burial type, or are derived by regular inhumations. And then we also see an interesting pattern when we again compare the damage rates to the dates of the samples. We see a different behavior between mummies and regular inhumations, where for mummies, there's actually a correlation with damage and age of the sample, and we're really not sure why that is the case, to be quite honest. And then lastly, we're seeing differences in damage rates depending on the lineage. Okay, so the clade that each ancient C. tetani genome is associating with, and that's what I'm going to get into next. Okay, so the next thing that we wanted to do is take all of these ancient draft genomes from C. tetani, and we wanted to build a phylogenetic tree, and we wanted to compare them with modern genomes. And this is really getting now more into looking at the evolutionary diversity and potentially the origin of some of these new genomes that we are identifying in ancient samples. Okay, so here on the left, this is depicting a phylogenetic tree of genomes. This includes all of the modern genomes and those are shown on the right in gray. These are non-bolded, and you can see that modern genomes fall into the two clades, clade A, clade 1 and clade 2. But interestingly, these new samples, these new genomes that we're identifying, they are not restricted to one lineage, but they're broadly distributed across the tree. We find several new genomes within clade A, and quite a few genomes within clade 2 as well. We also find several new genomes that cluster outside of the entire C. tetani tree, and we've labeled those genomes, or those that clade X. Okay, this is a new clade, and you can see based on average nucleotide identity in this heat map here that these genomes are quite distinct from those in the C. tetani clade, and they are at a level of genome identity that's below the same species level. So we actually think this represents a new species of Clostridium, and we'll tell you more, or I'll tell you more about that in a couple slides. One of the interesting things is that for some lineages, for example, lineage 1H, we've gone from one modern reference genome here to add 8 additional genomes from our ancient samples. So we're really expanding the known genomic diversity of some of these lineages. And now I'm just going to focus on a couple of these. So as I just mentioned, lineage 1H was expanded quite substantially from one reference genome from a modern sample to quite a few additional ones that we're identifying. And one of the interesting things is that these samples are geographically are non-randomly distributed. Okay, these most of these samples were coming from the Americas and from South America. These actually include the Chinchorro mummy samples that contain high levels of C. tetani. Another group that I want to focus on is this one here, clade X. Okay, clade X is quite mysterious to us, and this group of genomes again displays only about between 85 and 90% average nucleotide identity to other C. tetani genomes, but they're all very closely related to one another, which suggests that they do form a monophyletic group. They have a common ancestor. And we've done a lot of work trying to compare these genomes to ones that are available in the current database, and we cannot find any other species, any other sequenced reference genome that is close to them and clarifies what the species is. So we think that this species that we are assembling and analyzing from ancient DNA is actually, so far anyway, unique to these ancient DNA samples. It has not been observed assembled in any modern samples so far. So whether it represents just a new related species to Clostridium, to Clostridium tetani, that may be an environmental microbe that's out there that hasn't been sequenced yet, or whether it represents an extinct species present in these ancient samples, we don't know the answer to that yet. And we're going to be doing follow-up work to try to understand more about this organism. One of the really interesting things is that most of these samples are geographically clustered within Europe. So there is a very interesting geographical pattern. They are temporally quite diverse, but geographically they seem to be largely restricted to these ancient samples from Europe. And so, again, we've called this Clostridium species X, which seems to be completely unique so far to archaeological samples. And there, I'm just showing you, again, the geographic distribution on a map. Now, so far, I've only really shown you sort of bioinformatic analyses of ancient DNA and phylogenetic analysis. What we really wanted to see was whether these ancient pathogens were indeed tetanus toxin producing Clostridium. That's what distinguishes the C. tetani associated with tetanus disease versus perhaps environmental strains that are maybe related but lack the toxin. Okay. And one of the things we also wanted to see was whether we could identify novel variants of tetanus toxin. As I mentioned to you, there's extremely limited variation between different tetanus toxin sequences in modern genomes. So we wanted to see if we could uncover some additional diversity within these ancient samples. So this is showing you a structural model of tetanus toxin. Okay, so tetanus toxin has these 4 domains. The LC, or the light chain here, which is shown in red, this is really the key metalloprotease domain, okay, that is responsible for snare cleavage. The other domains are responsible for binding to human cells and translocation of the toxin into the cytosol. Okay. And so what we did was we looked at the tetanus toxin sequences within our samples, and specifically we wanted to see if we could find unique substitutions or SNPs that were completely unique to the ancient samples and were not observed in any modern tetanus toxin sequence. And so what you can see here is that some of these tetanus toxins that we've reconstructed from ancient samples show no unique variation compared to modern tetanus toxins at all. For example, these ones up at the top, they don't contain any unique SNPs. However, interestingly, there is a group of toxin sequences here. This is, I've labeled it as group 2 within this phylogenetic tree. These seem to contain quite a number of unique SNPs. And not only that, the SNP pattern, the profile is itself very similar between these three sequences. And one of them is noteworthy because this is actually the toxin that came from the oldest sample in our data set from the Chinchorro mummy. On the right, what I'm doing, I'm plotting the frequency of unique SNPs that are found within these toxins. So this kind of gives an idea of how unique, how different are these tetanus toxins from modern sequences. And this lineage of toxins, all of which come from ancient samples, really seems to stand out in that regard. So there's something unique about these toxins that are found within that lineage 1H from South America that I showed you a couple slides back. What you can also then do is you can take these SNPs and you can see which SNPs result in amino acid substitutions, and you can plot the location of those substitutions on the tetanus neurotoxin structure. And we also we see an interesting pattern where a lot of these mutations seem to be clustered on one external face. So a surface exposed region of the tetanus neurotoxin structure, that almost seems to suggest maybe there's some kind of relaxed evolutionary selection happening here, or maybe there's been positive selection for a change in binding preferences, maybe to an interacting protein. We're not sure what it is, but we find it interesting that a lot of these ancient substitutions unique to this lineage seem to be so clustered. Okay, and not only that, but some of these substitutions are shared then between different ancient toxins that we've reconstructed. So here I'm just showing you a segment, okay, from modern tetanus toxin here, and then below I'm showing you the same segment from two of the ancient toxins that we've reconstructed from ancient DNA, and you can see that they share several substitutions, again, that are completely unique to these ancient samples. These ancient toxins, they come from samples that are, again, geographically quite close to one another. Okay, and so we think that further supports the idea that these indeed represent a unique lineage of C. tetani or neurotoxin, and that they probably share a common ancestor. You can also do damage analysis of the toxin DNA itself, and we do find some evidence for damage in the toxin DNA. It seems to be similar to the damage profile that we observe for that genome overall, and this is really focusing on the Chinchorro mummy sample. And we wanted to focus more on the Chinchorro mummy sample because its toxin was perfectly reconstructed. We could actually reconstruct 100% of the toxin sequence. Now, this then led to the question: well, is this a genuine toxin? Are the SNPs that we're identifying real, or are they potentially random errors that have accumulated in these sequences? Well, one of the ways of testing that is to look for actual toxicity in the full-length protein itself. So to actually do assays to measure whether that protein is functional or not. Okay, and so this was done in collaboration with an expert at Boston Children's, that's the Mindong, also at Harvard. And Mindong's lab specializes in the testing of botulinum and tetanus neurotoxins, and they've developed a special technique for doing that avoids a lot of the safety or biosafety concerns that would result from full recombinant expression of these toxins. Okay, and this was especially important given that we were identifying potentially an ancient variant of a modern toxin, and so we wanted to do this in the safest way possible, and that's using this method called sortase-mediated ligation. Okay, where two separate halves of the toxin split into two components separately, and those components are then expressed and purified, and then they are so alone, when they are expressed and purified, they have no toxicity independently of one another, but when they are ligated together in the test tube, okay, through sortase-mediated ligation, they then form the full-length toxin that you can then test using various biochemical methods. Okay, so this was how the testing was done, and the tetanus neurotoxin from the chinchorro sample, we've called that tent chinchorro, was then tested to see whether it has the same function as modern tetanus toxin, and the short answer is that it was. It showed all of the same features of modern tetanus toxin. This blot here shows the ability of the chinchorro toxin here, labeled CH, to cleave the canonical substrate of tetanus toxin, that's VAMP2. This is one of the key snare proteins that are targeted by tetanus neurotoxins. So you can see the disappearance of the VAMP2 band due to proteolysis. Okay, so at a molecular level, it appears to have the same molecular phenotype or activity, but then also this was conducted on mice. So tetanus assays were done to look for evidence of paralysis in the mouse limb, and this is called a digit abduction assay. And again, the chinchorro toxin showed the exact same level of activity, or I should say the same phenotype as the wild type toxin. This was then quantified as well, and this ancient variant, or I should say this unique tetanus neurotoxin variant, we're not necessarily claiming that it is ancient, but this unique variant showed just as much toxicity as the modern form, and so it has extreme potency. One of the additional things I should say is that we looked, or my collaborators looked into the biosafety aspects in a lot of depth as well, and confirmed that antibodies to modern tetanus toxin were also capable of recognizing this variant. So it was it's very similar. There's only about a 1% amino acid difference overall, so it essentially is the same toxin as modern form. So that reduces concerns over several biosafety concerns that one might have. That concludes the work that we've done on this so far. Just to summarize, we've identified neurotoxigenic Clostridia in ancient DNA in 38 different samples. And this is really the first identification of these organisms in ancient archaeological samples. This also led to the discovery of a new Clostridium species, and we're going to be following up on this in other papers to come. And this new clade of Clostridium really appears to be completely unique so far, anyway, to ancient samples, and it's a fascinating finding, I think. We've also identified novel variants of tetanus neurotoxin. The one that I showed you seems to be unique to these ancient South American archaeological samples, and we've studied one of those experimentally. That's the tetanus neurotoxin from Chinchorro, which really appears to function just like the modern reference wild type toxin, and it exhibits extreme toxicity and also appears to cause tetanus disease in mice. If you're interested in more details, please check out our paper here. And I'd also like to acknowledge my lab for their involvement in this work, and also a number of collaborators from Harvard, Denmark, is Gabriel Renaud, my collaborator in anthropology at Waterloo, Alexis Dolphin, and Anna Dugan at McMaster Ancient DNA Center. And with that, I'll take questions. Thanks so much.
Thank you, Andrew. Definitely, it's a very intriguing and gripping work, you know, and that's why I might have too many questions, so I've trimmed them down, because I found, the whole study rationale very interesting. I think the work you presented today really sends the message that evolution or evolutionary biology is not only a historical science, but it also has implications, to modern life. I mean, I do hope that, any biologist would understand and appreciate that. But one of the goals of the series really is to promote evolutionary biology to everybody who has access to the internet. So hopefully someone who is not very acquainted with evolution will really appreciate that idea that, yes, we can perhaps, if I may say, even resurrect certain toxins, and there are, real world implications from the studies of evolution. Now, as I said, I have a few questions here. So when when discussing evolution of prokaryotes, I think the first thing that comes to my mind is pangenomes, right? We know prokaryotes, there is rampant horizontal gene transfer between closely related species and distantly related species. And I was thinking about, I was wondering whether, the concept of pangenomes would be an extra layer of challenge to your analysis, or you were able to factor that in, especially when you were talking about the clade X.
Yep.
I was wondering, what if this is just a sample of the pangenome?
Yeah, that's a great question. And that is actually something we explored, particularly for clade X. So one of the things that we did for the bulk of the analysis, the phylogenetic analysis and everything, is really restrict things to the core genome, not to the pangenome, right? We looked at conserved genes across the chromosome, in particular to do our phylogenomic work, right? So that provides that scaffold. You can still get an enormous amount of information looking at that core genome and building a tree. Where the pangenome came in was, well, for cladex in particular, if it really does represent a unique species, which the data suggested, right? Analysis of the core genome, the conserved genes, suggested that cladex was a unique species from Clostridium tetani. If that's true, we might also be able to find unique genes within the pangenome of cladex. And so we looked for that, and we actually so we assembled the full metaproteomes from the samples, and we looked to see whether we could find genes that were only found in those cladex samples and absent from the others, right? And we found many of those. Actually, we found, and we even went as far to show that they really were part of clade X and not maybe some other contaminating microbe, or maybe some other co-occurring species found in the same sample. We actually showed at a genome level that some of those unique genes had been inserted specifically within the genome of clade X, and that you could see that in Clostridium tetani that inserted gene was not there, right? So you could actually see at a genome level that a potential lateral gene transfer, for example, an insertion may have taken place, or the other possibility that it's been deleted uniquely within C. tetani. So, but you're right, like it's a big challenge, and of course there's going to be a lot of DNA there that's part of the pangenome. Maybe these are short fragments that cannot be assembled properly and linked to their parent organism, or linked to the core genome, and that remains a challenge, I think, for bioinformatics and in general.
Yeah, I agree, especially because the concept of the boundaries between a core pangenome, well, the core genome and the accessory genome, it's a moving target, right? The more strains you sequence, the core genome should shrink, and the accessory genome should change, let's say.
Yeah.
But again, great discussions. Now, well, I must say that I really liked that your work was based on publicly available data. This is what I do as well. I mean, I don't study ancient DNA, but my data come from SR8 only, so that was really nice to see.
Right.
And so in terms of having access to this data now, I was thinking, and especially when you mentioned, certain parameters that you were able to analyze and compare depending on the burial techniques, right? Whether it was mummification or not.
Yeah.
Would you see, down the road, would you see a possible analysis as looking a microbiome, so looking at microbiome differences depending on burial type? So let's say, perhaps with this data that are available out there in SRA, someone could determine whether different mummification techniques in different places, not only the geographic, but also time places, would, conserve bodies better or worse because they changed the microbiomes there?
Yeah, I mean,
I
think that's a great idea. And that's actually, that could be done. I think there are, given the data set we looked at and also other DNA, studies that have been done, those could be combined together, curated, right, to group them into, based on burial practices and things like that, and then that kind of analysis could potentially be done. I should say though that that was a challenging aspect of the study, because that data was not there as metadata within the SRA, for example. That was something that our anthropologist collaborators had to really help us with, to go into the original studies and categorize them based on these different types of burial contexts, right? That's not something we have any expertise in, to be
Mhm.
honest, right? So I see there's so much metadata annotation that can be done, um, that hasn't been done for the vast majority of samples in the SRA, but could give us all kinds of insights if
Mhm.
we could correlate genomic or metagenomic traits, right, with that metadata. I think there's so much potential.
I have the very same issue as well with data sets that are, poorly described and annotated in terms of metadata. I agree with you. It's
Yeah, yeah.
it's a challenge.
Right.
Now, another conceptual question. So, as you mentioned very early on, to determine whether sequences are ancient or not, you were looking at specific signatures at the end of the fragments, and that makes perfect sense to me. But I was wondering if C. tetani and related species, they
Mhm.
are spore forming species, right?
That's right. That's right.
So if we're looking for ancient DNA from these species,
Yeah.
and we find those signatures, does that mean those sequences, so they were environmental DNA, meaning that the spore exploded and they were lying there for millennia,
Hmm. Hmm.
or they are coming from spores and then they should
Yep.
have, they should be more conserved?
Yeah. I mean, this is something I don't have a great grasp on, to be
Mm.
honest. Is the effect that being a spore former has on observed rates of DNA damage, and how that relates to the time of the sample, right? Like that's actually a question I have, and I've tried to investigate that by talking to a lot of people in the field, and um I don't have a clear understanding of that.
Mm.
Um the but it is entirely possible, yes, that the DNA that ends up getting sequenced is um released DNA from lysis of these spores. Maybe that DNA alone has been sitting in these samples, accumulating damage, not protected in a spore form, right, for a certain amount of time, like that. Um, some of it, some of the DNA may be protected by spores, being in a spore state, and some of it may not, and we may be observing a mixture of those two signals, right? Um, but I think, yeah, this you're kind of touching on one of the biggest issues that we have, when just taking an ancient sample like this and doing a metagenomic analysis and then profiling the damage levels. Just because we get a certain level of damage or not, that doesn't necessarily tell us anything about the precise dating or the origin of the DNA. Now, it's different if you're studying a pathogen, um, like say Mycobacterium tuberculosis or plague, and you have other data available for your archaeological sample that implicates that microbe in the cause of death, right? So it's then any DNA that you identify that appears damaged is additional evidence, right? Becomes additional evidence of an ancient infection. In our case, it's very different. We did a broad metagenomic survey, and then so I think it's very likely that at least for a subset of our samples, that these may just be some of it might be ancient contamination, or maybe not, maybe that's the wrong word, but ancient colonization of human tissues by environmental clostridial pathogens. That is entirely possible, but the thing is that's still of great interest to many of us in the field, because we can still use that to explore genomic diversity and evolution and look at variants of different virulence factors and find new species. Like it, our reason for doing this study was not to try to find a new cause of death in ancient sample archaeological specimens. Yeah.
Wonderful. All of that makes sense to me. Well, Andrew, I think I've already asked you too many questions, so I guess we can wrap things up here. I will stop the recording in a second, but then you and I, we can just chat for a minute or so before I let you go. But again, thank you very much for the very insightful talk. And I'm pretty sure the audience, whoever will watch this, will appreciate the insights and the conclusions we all came together. Thank you, Andrew.
Thank you.
I will stop recording now.